So today I'm here to talk about technologies to address the global catastrophic biological risks as we go into this field that is relatively new in Africa. Okay, next slide, please. Okay, so there are various technologies that we require in order to address the global catastrophic risks. So these technologies are majorly in two different sections. We have technologies for disease detection, surveillance, and situational awareness. We also have technologies for infectious disease diagnostics. We have technology for distributed medical countermeasure manufacturing. We also have technologies for medical countermeasure distribution, dispensing, and administration. And in the case of a global catastrophic biological risk, we should be able to have technologies that can address medical care and address surge capacity in the medical system and the medical institutions, just like we saw with the COVID-19. Next slide, please. Okay, so for the technologies for disease detection, surveillance, and situational awareness, the first thing is the ubiquitous genomic sequencing and sensing. So with genomic sequencing and sensing, we have improved detection of the functions of these pathogens that are causing these risks that we have. Okay, so we need to invest in technology for genomic sequencing, the bioinformatics, the analysis, so that we are able to detect the pathogens faster, because in the event of a global catastrophic biological risk, the spread is pretty much fast. So we need these technologies for sensing and genomic sequencing so that we are able to address this challenge as fast as we can. Now, the analysis of information, genomic sequencing, and the data that's usually generated from these processes, the data is usually in large volumes. Okay, so we need technologies that can overcome the bottlenecks of storage, interpretation, and the analysis of genomic data, omics data generated from the sequencing. Now, the challenge with adopting such technology that we have, especially in Africa, is that we lack sufficient skills to perform analysis, even the sample preparation. Now, even when we perform the analysis, interpretation is usually an issue. Okay, and we don't have enough technology to have this in place. So if we are going to prepare ourselves for addressing these global catastrophic risks, then we need to invest a lot in technologies that will address sequencing and remote sensing. Next slide, please. Okay, so we can also adopt drone networks for environmental detection in an effort to address the GCBRs. So for drones, or the drone networks, which have not yet been much adopted in Africa, we could use them to collect air and water samples, monitor, events that are happening before they happen, predict them, so we can avoid catastrophic changes to environment. And then sometimes it's difficult to also access contaminated areas, but if we need samples so that we can get information, do the analysis, understand the pathogens, then we need to be able to access those contaminated areas collect samples, do the analysis, so we can make sense of what is happening and how fast the pathogens can spread. And so the drones can also be used for situation analysis and awareness. However, in Africa, the technology is relatively new. So for us to be able to adopt this particular technology here, we also need policies, okay, that govern how we use drones so that we are able to use them for exactly what we want. If we are using drones for detection and collecting samples and situational awareness, we should regulate their use so that they're not used to collect information that would otherwise, okay, draw back our effort to mitigate global catastrophic biological risks. Next slide, please. Okay, so another technology that we need to invest in is remote sensing for agricultural pathogens. Okay, so remote sensing will enable us to be able to detect disease symptoms early, sometimes before they actually occur. And basically, we can use it in agriculture to avoid spread of pathogens across people's farms and, on a large scale. We can mitigate that before it actually goes onto a large scale. Next slide, please. Okay, so as we said earlier, we also need to invest in technologies that will be used for infectious disease diagnostics, okay? Now, the first technology that we need to adopt on a large scale, especially in Africa, is the microfluidic devices. So these devices here, okay, they use minimum fluid. They're very fast in making diagnosis on the bedside, okay? So in the event of a global catastrophic biological risk, we need devices that can be used to detect the diseases as fast as possible, okay? As fast as possible. So these devices can be used for diagnosing infectious diseases, especially in the field, okay? Like HIV, hepatitis V, Zika viruses, and etc. They have actually been used before, okay? Now, these devices here are known to produce high accuracy, they're easy to use, and a shorter time for obtaining results, which is what we usually want to have in the event of a global catastrophic biological risk. And it also comes with lower costs. So because it comes with lower costs, it's much easier to adopt on a larger scale. And so it is very useful if we can adopt this kind of technology on a wide scale so that we avoid, we curb the impact and effect of a global catastrophic biological risk. Next slide, please. The second technology that we need to adopt for diagnosing infectious diseases is handheld mass spectrometry. So this one also is much easier to use. And in the event of a risk, a global catastrophic biological risk, health officials can actually use this to rapidly identify the affected population. So because these machines are portable, they're easy to use, they're fast to use, and you can mitigate the effect of a global catastrophic biological risk in the shortest time possible, okay? Next slide, please. The other technology that is important to adopt is what we call the cell-free diagnostics. So here we usually have engineered genetic circuits to bring lab into the field, using freeze-dried cells, especially for the lab people, they understand that. So this approach also is important because it allows diagnostics in a much faster manner than the conventional lab diagnostics, which would take a longer time. Now, in the event of global catastrophic biological risks, we are usually running against time to curb spread and therefore prevent too much damage to the society. Next, please. Okay, so aside from technologies that are used to diagnose infectious diseases, we also have technologies that we need to adopt for distributed medical countermeasure manufacturing. Now, this is important because in the event of a global catastrophic biological risk, we need to be able to, produce drugs in a mass form, and the vaccines as well. As we've seen with the COVID-19, the vaccines were able to come up in a faster form than they would otherwise do. So one important thing that we need here is synthetic biology for manufacturing medical countermeasures. Okay, so with synthetic biology, we can engineer the strains of the organisms. We produce the drugs and the vaccines very fast, and we should be able to deliver these to the society or to the affected areas in the shortest time possible. Now, with this, we need to invest much earlier before the global catastrophic biological risk occurs, okay? Because once it occurs, then we don't have time to invest in the technology at that time. So if the technology already exists, then we go ahead to produce drugs and vaccines that could reduce the spread of diseases within the community, just like we saw with COVID-19. Next. Okay, we also have three-dimensional printing of chemicals and biological structures. So this is very important because it helps us to actually structure our medication, okay, into different formats and customize them to the patients, to the disease. And in that way, we have better outcomes for treatments, for the risk, depending on the risk that we are addressing at that point. So because of 3D printing, even drugs, okay, can be manufactured in better dosage forms, and then the delivery systems can also be improved so that we deliver the medication to the affected population in the shortest time possible. Okay, but this kind of technology needs to be adapted earlier in preparation for global catastrophic biological risks that may occur. Okay, so in Africa, we haven't yet really gone into accessing such technology. So we encourage us and policymakers, okay, to go ahead and put this across, but also we need structures through which we can do these things. Next slide, please. Now, aside from those technologies, we also have technologies for the medical countermeasure distribution, dispensing, and administration. Now, these technologies are very important because they can be easy to address the risk in case it happens. So we're going to start with the first one, the microarray patches for vaccine administration. Now, microarray patches for vaccine administration is a fairly new technology in which the vaccines can be administered basically without needles, okay, and also eliminating the cold chain for transporting these vaccines. So with such a technology, it makes it easier for the vaccine to be delivered to as many people as possible in the shortest time possible. However, we need to create more awareness for such microarray patches of vaccine administration so that it is more acceptable within the community. Because if the community does not accept this on a large scale, then the technology cannot be adopted to, mitigate global catastrophic biological risks. The second technology that we can adopt for this is self-spreading vaccines, okay? Now, self-spreading vaccines are these vaccines which are inoculated into the host, and just like the pathogen spreads within, the self-spreading vaccine can also spread within, Okay, now these self-spreading vaccines usually are good for diseases that are transmitted from animals to humans. So if they can be curbed within the animal population so that the vaccine can spread within, then we avoid situations where they can be easily transferred from the wild animals to the humans. So this kind of vaccine that is self-spreading, this technology should be adopted on a wide scale, okay? Especially, so in the recent past, there have been developments for self-spreading vaccines for Ebola, for the Lassa fever in West Africa, and many others. And then we have the ingestible bacteria for vaccination. This can be given to people through plants, okay? So this kind of vaccine is introduced genetically into plants, but with preserving their power, their immunology power. And then if you ingest that, you should be able to receive the protective effect. However, still, the population needs to be enlightened on how this works so that they're able to adopt such kind of technology as well. And of course, in Africa, we need more training so that we are able to actually do the genetic engineering so that we can adopt vaccination through ingestion of plants. And then we also have synthetic vaccinology, self-amplifying mRNA vaccines. These, have a shorter turnaround time. And lastly, we have, in a situation where we already have a global catastrophic biological risk, we may need drone delivery to remote locations, of the vaccines. However, before we take on drone delivery to remote locations, you need to assess whether it is feasible under the circumstances and how much the effect of the risk is at that point. Next, please. Okay, and then lastly, we also have technologies for medical care and surge capacity, especially in the event that the global catastrophic risk is already in place, it's already taking place. So what you need there is to be able to actually improve capacity to handle the number of cases that may be available. So for example, in the COVID-19, portable, easy to use ventilators. Now there's so many technologies under this, but we'll talk about maybe one or two. So we also have robotics and telehealth to reduce congestion within the health facilities and many other technologies that we may eventually adapt in case we are faced with a global catastrophic biological risks. Next. Yeah, so we have a few references. And next. Thank you for listening.
Thank you very much, Ms. Paece Aber, for that very brilliant presentation. I'm a scientist, but I've learned a whole lot of new things today. So you have the first question from Tim here.
Thanks for a wonderful overview of the new technologies coming our way. One question I had, the microfluidic devices, how long will it take to produce these for a novel agent? So for example, we had COVID appear in the world sometime in late 2019. How long would it take from first characterization of the virus to actually being able to produce these devices in mass quantities?
Well, thank you for that question. I'd like to defer this question to the lab specialists.
Thank you for these presentations and this indeed informative presentation regarding the new technology. My question also regarding the microfluidic device. You say that they will be more efficient in cost, time, and accuracy. So my questions for the the accuracy. Do you know exactly what is the sensitivity and specificity of this test, and the degree, or what is it highly in specificity and highly in sensitivity, or in both, or only one? Because it is very important to determine how we can functionalize or operationalize this device in the field or in the labs. Thank you.
Thank you.
Thank you very much for that question. I think we have two papers that have published that, so maybe I will refer to those papers. So I think on the path of sensitivity, it's not very high, but specificity is very high. So if I still remember that paper, but I will send you the paper. Okay.
Thank you. You have another?
Thank you for your presentation. I would like to have more information about cell speed vaccination, about for Lassa, for example. I would like to better understand the technology and if it is available or if it is a project.
Okay, so that was also published because the work was Ebola, I think, came to West Africa some times back. And the paper was published on that, and I will also send you the paper on that, okay?
Thank you, Stephen. I'm still curious to know more about that vaccine, the self-spreading. Could you be knowing how efficient and effective that vaccine is?
Yeah, that's also a good question. If I remember from the paper that we read, I think it was very effective when they did the trial. But again, I will send you the paper so that you can go through. So most of this presentation that was presented by Paece have been published. That's why she put all these references there. Most of them are just new technologies.
Thank you. We have one more question.
Thank you. I seek clarity for the ingestible
Hold on.
bacteria.
This is a new participant, so she'll introduce herself, name.
And I'm coming from Uganda. You're welcome. I seek clarity about the ingestible bacteria for vaccination. At what stage are you introducing them into the plants? Are you waiting for the plants to grow, or because I'm looking at the catastrophes as something that happens rapidly? If we have to wait for the plants to grow, then that would take a little bit longer. Then is it at harvesting stage that you'll have to introduce into the produce or something? Thank you.
So vaccine have been produced in plants by very many organizations. It depends on the vaccine. So for global catastrophic biological risk, I think what they have to do is maybe produce the vaccine and maybe keep it in the form of a seed or something that can be easily, for example, the seed. Then if the catastrophe comes, then the seed can be maybe grown or can be crushed, and then the vaccine can be got. But vaccine have been produced in plants by very many organizations. Depends on which stage.
So there's an assumption that probably the pathogen that could bring about a global catastrophe hasn't yet been encountered. So there could be no vaccines in storage. In such a scenario,
that's why you need to plan ahead. You need to plan ahead. Even for COVID-19, COVID-19 vaccine came because the work, somebody had already been doing the work before, by the way. That's why when COVID-19 came, they had to go back and get that information, and that's why. So we have to start working on that now.
Thank you. Any other questions, physical or online? I'll take this and see if there are any hands online. Those are online, you just have to put up your hand, and I'll select you to unmute and ask your question.
Okay. My question is, you stated it when you have new technology, mainly people refuse it because it's something that is new. And another challenge is in Africa, you have most of the people have limited resources. So when you combine those two things, how do we think this new technology can help African country about GCA, global biorisk?
If they can be useful in Africa. So the technology is new, adaptation is low, but if we can invest in the new technology and create awareness, then they should be useful to Africa. But for now, where we have technologies that are very new to Africa, we don't have the skills to use them. But since we are preparing ourselves, okay, to mitigate the global catastrophic biological risk when it occurs, this is the time to invest in the technology. This is the time to invest in that.
And to address your question of affordability, I think one of the key things that these new technologies potentially offer is that they will be cheaper. So, for example, if you're using microfluidics, rather than having to do in-laboratory analysis, that's a lot cheaper. The overhead is much reduced because you don't have to have an expensive BSL-3 lab to do it. You've got handheld pieces of equipment that are mass-produced and are much cheaper than the old way of doing the analysis. The second is that they reduce the skill level required to do the work. You don't need PhDs to use a handheld device that has built into it the software to do the interpretation. So I think in both of those ways, in reducing, or 3 ways, the unit cost will come down, so it will be more affordable. Still will be an issue with resource challenged countries, but it will be more affordable than what currently exists. The overhead will be much reduced and the skill requirements will be reduced.
Thank you very much, team, Dr. Stephen and Ms. Paece for that. There's a hand online. David, you can unmute and ask the question.
Okay. Thank you very much. I'm David Mtweve, a master's student at Busitema University. So first of all, thank you very much for the comprehensive presentation from Madam. Secondly, my question or worry was on the technology. Looks to be super good, but our countries ready to provide like subsidies or like some fairness to the researches that are going on, especially in that area, because I have experience that I was working with one of the projects and making one of the portable technology for detection of aflatoxins. Now, there had been a lot of issues. You order, let's say, antibodies from the UK. The antibodies are subjected to the VAT, and then the clearance agents will tax you or give you a lot of money to pay. So are our countries ready to adopt the technologies? And if yes or no, maybe the organization would find a way to find a way to reduce the cost for the researchers, especially in the African context. If we can have these handheld technologies for responding to the catastrophes, which will be of very high value to the countries. But I don't get the point if our countries really understand the sensitivity of the thing, because they might have the cutoff that if you order something from the outside, this is the value added tax you have to pay, no matter what you are doing. Yeah, so that was my input. Thank you.
Thank you very much, David. I understand you. And I'm going to, first of all, I think we need policy and maybe legal framework, especially people in the government, that can help to see the advantages of some of these technologies. And then secondly, I live in the US. but I do both work in US and also in Africa. I'm going to give you an example. We have a bioplastic company based in Zimbabwe. So we always order reagents from either US or UK. So here's reality when you live in Africa. Sometimes when my colleagues try to order some of these reagents, they don't even reply their emails. But if I use my US email account, they reply those emails. So I think the government and the policymakers should try to help us, people who are in Africa, to break that barrier. So it starts with the policy, and then also to educate them that what we are trying to do is going to help the continent. Thank you.